Hello viewers, good morning. Today let us discuss on a topic related to molecular biology, that's regulation of gene expression in eukaryotes. Now before going to the actual regulation process, let's understand where exactly the gene gets expressed. Now gene expression is a combined process of the transcription of the gene into mRNA and the process of that mRNA or the processing of that mRNA and its translation into proteins for protein encoding genes. Now we should understand there are various levels of regulation of gene expression. That's gene on DNA gets transcribed into primary transcript and then further mRNA, finally to protein, the central dogma of molecular biology. Now, there are various levels of regulations the gene undergoes. One is at transcriptional control, RNA processing control, RNA transport control, and translation control. Now, what is the purpose of regulation of gene expression? When a gene is expressing itself, why there need to be a regulation on the expression? Now, regulation expression of genes is required for various purposes. The first one is adaptation. The cells of multicellular organisms respond to varying conditions. Such cells exposed to hormones and growth factors change substantially in their shape, growth rate, and other characters. Now, the second purpose is tissue specific differentiation and development. Now, the genetic information present in each somatic cell of an organism is practically identical. Cells from muscle and nerve tissue show strikingly different morphologies and other properties, yet they can exactly contain or they have exactly the same DNA. So these drivers properties are the result of differences in the gene expression. So expression of the genetic information is regulated during ontogeny and differentiation of the organism and its cellular components. Mammalian cells possess about 1,000 times more genetic information than that of a bacterium like E. coli. Now, mechanism of regulation of gene expression, let's have an overview. Gene activity is controlled first and foremost at the level of transcription itself. Much of this control is achieved through interplay between the protein that bind to the specific DNA sequences and their DNA binding sites. This can have a positive or negative effect on transcription. Accordingly, you have positive control and negative control mechanism. Now, transcription control can result in tissue-specific gene expression. In addition to transcription level controls, gene expression can be modulated by gene rearrangement, gene amplification, post-transcriptional modification, and RNA stabilization process. Now, there is differences between gene expression in prokaryotes and eukaryotes. Gene regulation is significantly more complex in eukaryotes than in prokaryotes for various reasons. The very first reason is the genome being regulated and is significantly larger. If you take E. coli, the genome consists of a single circular chromosome. This genome encodes approximately 2000 proteins. But in case of eukaryotes, the genome is larger. That is, it contains 23 pairs of chromosome, which ranges from 50 to 250 MB. Approximately 40,000 genes are present in human. It would be very difficult for a DNA binding protein to recognize a unique site in this vast array of DNA sequences. So more elaborate mechanisms are required to achieve specificity. Different cell types. Now, different cell types are present in most eukaryotes, like liver cell, kidney cell, pancreatic cell, for example, differ dramatically in genes that are highly expressed in them. The different mechanisms are involved in regulation of such genes. One difference is absence of operons. In eukaryotic genes, or the eukaryotic genes are not generally organized into operons, or as in case of prokaryotes. Instead, genes they encode proteins for steps within a given pathway, or often they are spread widely across the genome. The chromatin structure. The DNA in eukaryotic cell is extensively folded, and they are packed into protein DNA complex. What do you call it as chromatin? Histones are an important part of this complex since they both form the structure known as nucleosomes and also contribute significantly into the gene regulatory mechanisms. The fifth difference, the uncoupled transcription and translation process. In case of prokaryotes, transcription and translation are coupled process. The primary transcript immediately is translated, but that is not the case in eukaryotes. They are uncoupled process. So the primary transcript in eukaryotes undergoes some modification before a mRNA gets matured into a functional RNA. Now, mechanisms of regulation of gene expressions can happen at different levels. You can have chromatin remodeling or chromosome remodeling. So here, chromatin structure provides an important level of control of gene expression, transcription. With few exceptions, each cell contains the same complement of gene. So, the development of the specialized organs or tissues and cells and their function in the intact organism depend on the different or differential expression of the genes. You have chromatin remodeling. For example, the DNA containing the globin gene cluster is active in chromatin in case of reticulocytes, but they are inactive in case of muscle cells. Next, we have formation and disruption of nucleosome structure. The presence of nucleosome and the complex of the histone and the DNA provides a barrier against the ready association of the transcription factors, which is required for the transcription process. So that is how they are regulated. So the disruption of nucleosome structure is therefore an important part of eukaryotic gene regulation. Next, we have enhancers and repressors. So enhancer elements are DNA sequences, although they have no promoter activity of their own, but they greatly increase the activities of many promoters in eukaryotes. So enhancers function by serving as binding sites for specific regulatory proteins. So an enhancer is effective only in specific cell types in which appropriate regulatory proteins are expressed. Now, these enhancer elements can exert their positive influence on transcription, even when separated by thousands of base pairs from a promoter. They work when oriented in either direction, whether it is upstream or downstream. So, enhancers are promiscuous. They can stimulate any promoter in the vicinity or they can act at far off places. So the elements that decrease the expression, you call it as repressors. So the expression of specific gene has been identified. So silencers are control regions of DNA that, like enhancers, may be located thousands of base pairs away from the gene they control. However, when transcription factors bind to them, expression of the gene are controlled, that is repressed. So tissue specific gene expression is mediated by enhancer or enhancer-like elements. So you understood there are two things, enhancers and repressor, which can control. Now the next thing you have is gene amplification. The gene product can be increased by increasing the number of genes available for transcription of a specific molecule. So among the repetitive DNA sequences are hundreds of copies of ribosomal RNA genes and tRNA genes. So during the early development of an organism, say metazoans, there is an abrupt increase in the need for ribosomal RNA and messenger RNA. So that makes up such organs, say as the eggshell. So such requirements are fulfilled by amplification of these genes. So subsequently, these amplified genes generated by the process of repeated initiation during DNA synthesis, it provides multiple sites of gene expression. Now, gene rearrangement is one more. Gene rearrangement is observed during immunoglobulin synthesis. So, when you have immunoglobulin synthesis, immunoglobulins are composed of two polypeptides. One is heavy chain, the other one is light chain. So, the mRNA which encodes these two proteins subunits are encoded by gene or gene sequences that are subjected to extensive DNA sequence coding changes. So these DNA coding changes are needed for generating the required recognition diversity for appropriate immune function. So the Ig molecule light chain is composed of variable, J chain, that's joining region, and C chain, the domains or the segments. For a particular subsets of Ig light chain, there are roughly 300 tandemly repeated variable gene coding segments. So 5 tandemly arranged J chains coding sequences and roughly 10 C genes coding segments. So all of these multiple distinct coding regions are located in the same region of the same chromosome. So however, a given functional IgG light chain transcription unit will contain only the coding sequence for a single protein. So thus, before a particular IgG light chain can be expressed, single V chain, variable, joining and constant coding sequence should be recombined to generate a single transcription unit which excludes the multiple non-utilized segments. Now, next you have alternative RNA processing. So, you know, eukaryotic cells also employ alternative RNA processing to control gene expression. This can result when alternative promoters, intron, exon, splice sites, or polyadenylation sites are used. So, occasionally, heterogeneity within a cell will result, but more commonly, the same primary transcript is processed differently in different tissues. So, alternative polyadenylation site in the immunoglobulin, heavy chain primary transcript results in mRNA that are either long base 2700 long base or 2400 base long. So this results in a different carboxyl terminal regions of the encoded proteins that and immunoglobulin secreted by the cell. So alternative splicing and processing will result in the formation of unique mRNAs in different tissues. Now next you have class switching. What is class switching? In this process, one gene is switched off and a closely related gene takes up the function. So during this process, one is on, the other one is off. So during the lifetime, embryonic hemoglobin is the first hemoglobin to be formed. It is produced by having two zeta and two epsilon chains. By the sixth month of the lifespan of the embryo, hemoglobin is replaced by hemoglobin F, which consists alpha chain and gamma chain. So after birth, HbF is replaced by the adult type hemoglobin A. Thus, the genes for a particular class of hemoglobins are switched off and for another class are switched on based on the need. So gene switching is also observed in formation of immunoglobulins. That is a reference to primary immunoglobulin or primary immune response, secondary immune response. You can also observe this kind of regulation, which also is observed in mRNA stability. If a mRNA should be stable after transcription process, you need to have 5-cap structure which is formed and a 3' tail. So you call this as capping and tailing process, without which the mRNA is not stable. What happens? It gets degraded. Otherwise, it is not functional. Now, let's look into the regulation process which occurs in galactose. Now, you know prokaryotes will get regulated by an operon concept. In eukaryotes, there are various levels of regulation. So, eukaryotic gene expression is regulated at 6 levels. One is transcription, next is RNA processing, mRNA transport, mRNA translation, mRNA degradation, protein degradation. So, at these levels, you can have the expression and regulation of the gene. Now, promoters actually are controlled. That's the transcriptional control of gene regulation. The first thing is promoters. Next, you have enhancers at that level. Then you have activation of transcription factors, which are also involved in the control mechanism. Now, let's look into the transcription control of galactose utilizing genes in yeast. Now, there are 3 genes, GAL1, GAL7, and GAL10, which codes enzymes that functions in the galactose metabolism pathway. So, GAL1 is responsible for the production of galactose. GAL7 is responsible for galactose transferase. GAL10 responsible for galactose epimerase. Now, the pathway produces D-glucose 6-phosphate, which enters a glycolytic pathway and it is metabolized further continuously for utilization of the galactose. So, in the absence of galactose, GAL genes are not transcribed. GAL genes rapidly is actually induced by galactose and absence of glucose. So, analogous to E. coli lack operon repression by glucose. Now, the final product of metabolizing or metabolism of the galactose is the glycolysis pathway. Now, how it is controlled? GAL genes are near each other but not constitute an operon. Additional unlinked genes, GAL4, codes for a repressor protein that binds to the promoter element called as upstream activator sequence. Now, upstream activator sequence is located between GAL1 and GAL10. Transcription occurs in both directions from upstream sequence. When galactose is absent, the GAL4 product, that's GAL4P, and another product, GAL80P, will bind to upstream sequence. So there, transcription doesn't occur. There's a blocking of the transcription process by GAL80. So it's a negative regulation. When galactose is added, a galactose metabolite will bind to GAL18 and GAL4P amino acids are phosphorylated. So now galactose will act as an inducer by causing a conformation change in the GAL4P and GAL4ET complex. Now you have activation model. It means when there is no galactose, there is no transcription occurring. So it's not getting activated. But in the presence of galactose, which can act as an inducer, you have GAL4P protein getting a conformation change and it leads for the transcription process. So here you can see two metabolic or two kinds of regulatory process. In the presence of galactose, you have transcription. In the absence of galactose, you don't have the process. So galactose becomes an inducer there. Now, what is the responsibility of the genes locator? You have GAL7 producing galactose transferase meant to transfer the galactose. Then you have epimerase, which will change the structure of galactose into a epimeric form. Then you have kinase, which is responsible for phosphorylation of the molecule. Now, this kind of regulation is also observed in hormones, which is a very good example where you have thyroid hormones getting regulated. Now, let's wind up the process. as to the importance of the summary of the regulation. Now, in case of prokaryotes, you have studied the control mechanisms in case of lacoperon as an example and tripoperon as an example, where you have regulation of transcription. But in case of eukaryotes, you have various regulatory processes which occurs at six different levels: transcription, RNA processing, mRNA transport, mRNA translation, mRNA degradation, and protein degradation. Thank you.